Hello students. In previous lectures, I tell you about the light reactions, about the different pigments. Today, I am going to tell you about the dark reactions, right? And another one is, here it is given chemi-osmotic hypothesis. It is important because how ATP is formed, it is given here, right? What is given here? Let us now try and understand. What we try and understand? How actually ATP is synthesized. How ATP is synthesized. We know we have two cell organelles in which ATP is formed. First one is our chloroplast and mitochondria. This first one chloroplast, another one is mitochondria. Mostly involved in mitochondria. In animal, both animal and plant cell, mitochondria is there where ATP is formed. Right? We know very well. And this is our chloroplast, means it is present in only plants where ATP is formed, but less in. Both are different, right? Because here photophosphorylation is there, and in mitochondria it is not a photophosphorylation, it is chemi-phosphorylation, right? We also said them chemophosphorylation. Phosphorylation means what? Formation of ATP. Photo means in the presence of light, and chemi means what? In the presence of chemicals, or sometimes we said them oxy, in the presence of oxygen, right? What happened here? What is given here? Because here we are going to read about the photosynthesis, and in photosynthesis it is used, means the energy which is used from light, so it is called photophosphorylation, right? How actually ATP is synthesized in the chloroplast. Look, ATP is synthesized in chloroplast in which part? In grana, in thallopides, right? The chemi-osmotic hypothesis. Chemi-osmotic means chemical, osmosis means what? Osmosis, it is a process in which substance from higher concentration moves to lower concentration, right? Has been put forward to explain the mechanism. Okay, we will read here. Like in respiration means in mitochondria. In photosynthesis too, ATP synthesis is linked to development of a proton gradient. Proton means H plus ion. We said them hydrogen ion is also called as proton. So concentration means two different concentration, that is called proton gradient across a membrane. Here we need what? We need proton gradient, one membrane, one proton, that is proton gradient. Third one is our athlete's proton pump, right? So these three is very important for formation of ATP, right? What is even? ATP synthesis is linked to development of the proton gradient across a membrane. This time these are membranes of thallopides. This time is in chloroplast. These are the membrane of thallopides. But in case of mitochondria, these are the membranes of inner mitochondrial membrane. It is inner mitochondrial membranes, or we can say these are the Christie membranes. There is one difference though. Here a proton accumulation is toward the inside of the membrane. That is the main difference. Here proton, what is given? Proton accumulates, means concentration of proton is present in the intermembrane space of lumen, right? That means inside the membrane, that is in lumen. But in case of mitochondria, what happens? Just opposite. Means proton gradient, or we can say concentration of proton is more toward the perimitochondrial matrix, that is outside the mitochondrial matrix, not inside but outside, right? In photosynthesis, what happens? In chloroplast, what happens? Proton accumulates inside the membrane. But in mitochondria, what happens? Proton accumulates outside the membrane. That is the difference. So that means what? Their moving direction is also different. In chloroplast, what happens? Proton moves from inside to outside. But in case of mitochondria, proton moves from outside to inside, right? In respiration, what happens? Proton accumulate in the intermembrane space. Intermembrane space means what? Outer and inner lamellae. Between outer and inner lamellae, space of mitochondria. when electron move through the ETS, electron transport system. Look here. Here what happens? Look, this is our chloroplast and this is our mitochondria. In mitochondria, we know these finger-like projection, they are called cristae. Here what happens? Protons are more. H plus ion concentration is more. And here H plus concentration, this is the less concentration. Less. And here concentration is, it is more concentrated. Right? Concentration is more. What happens here? The direction of flow of hydrogen is from outside to inside the mitochondrial matrix. That is the direction, right? But in case of chloroplast, here thylakoids are. These are thylakoids. And in between this space, this is called lumen. What happens here? In this lumen, H plus concentration is more. And outside, H plus concentration is less. That means what happens? Proton moves from inside to outside. That is the main difference, right? Let us consider or understand what causes the proton gradient across the membrane. How a proton gradient is formed inside in the membrane, right? We need to consider again the process that take place during the activation of electron and their transport to determine the steps cause a proton gradient. Here what happens? We know here also electron transport system is there. Maybe it is cyclic or non-cyclic, it does not matter, right? But here electron transport system is also responsible for the accumulation of hydrogen ion, right? Look here, 3 steps are there. When we see these 3 steps, we can understand how the more hydrogen atom they accumulate inside the lumen, right? First what happens in hydrolysis? What happens? Look here. What is given? Then I tell you. Since splitting of water molecule take place in the inner side of the lumen, inner side of the membrane means lumen, right? Water splitting occurs inside the lumen. What happens? When water splits, it breaks into H plus and OH minus. H plus and it accumulates inside the lumen, right? And this OH means it binds with another OH and forms one H2O molecule, and oxygen gas is released. That means it goes outside the chloroplast, right? This H plus ion, it accumulates inside the lumen. First step. Second, when electron transport system is there, what happens? When electron, which is come from this hydrolysis, right? It is transported to the first electron acceptor. Who is first electron acceptor? It is pheophytin, right? Okay, I tell you here. First we read this and then I will explain everything. See, first step, I already tell you. Since splitting of water molecule take place in the inner side of the membrane. Where? Inner side of the membrane, that is inside the lumen, right? The proton, that is hydrogen ion, that are produced by the splitting of water, means look here, H2O, now it produces H plus and OH minus, right? So this H plus, it is accumulated inside the lumen. First thing, what happens for this OH minus plus OH minus, it forms H2O and plus half O2, we can say, and this half O2, it goes out of the chloroplast. Now one H2 is formed. Now this OH minus, this is not present because they forms a water molecule and oxygen gas. This oxygen gas removed, that is left from the that is lumen, and 2 electrons are formed. 1 electron and 2 electrons. These electrons, they come into the electron transport system. Then second step start, right? When these electrons, so these are transported to pheophytin, which is first electron acceptor. This first electron acceptor, it transports this electron to the hydrogen donor, right? Hydrogen acceptor. Now this hydrogen donor, it remove 1 hydrogen from stroma and took it to the, that means it also release this hydrogen atom which is removed from stroma. It is left to the lumen, means 1 H plus ion also added here, 1 from water molecule, splitting of water, another from stroma to lumen through this electron transport system, and third means it is not come inside the lumen, but two H plus ions are removed in the formation of NADPH2, right? That's why three hydrogen atoms, they are removed from stroma, right? So in which two is involved in the formation of NH2 and one is that means transfer from stroma to lumen, right? That means what, 1 minus 1 is that is minus from the stroma, but it is added into inside the lumen, and one another H plus ion, it comes from where? It is through water splitting. So in lumen what happens? It is a continuous process. So after some time what happens? More and more H plus ion stored inside the lumen. Then what happens? If more H plus ion are inside the water, inside the lumen, then what happens? Inside the lumen, pH value is decreased. Means it becomes acidic. Okay. Okay. Look here. What is given? So first you understand this one, splitting of water. Second is given here. An electron moves. As electron moves through this, which electron? The two electrons, which come from water splitting, right? Which come from water splitting. Splitting. These two electrons, which come from water splitting, right? They move through the photosystem, that is through electron transport system. Protons are transported across the membrane. This happens because the primary acceptor, who is primary acceptor? It is our pheophytin. Simply we write them PHE, right? Pheophytin. The primary acceptor of electron, which is located toward the outer side. What is the location of this one? Toward the outer side of the membrane. Transfer its electron. Electron means this electron, right? Not to an electron carrier, but to an hydrogen carrier. Pheophytin. This pheophytin, it transports its electron not to a hydrogen carrier, but to a electron carrier, but to a hydrogen carrier, and this hydrogen carrier is our blastoquinone. We simply write them PQ, right? Hence, this molecule removes a proton from the stroma, and this PQ, it removes a H plus ion from stroma. This side stroma is there, right? Then what happens? While transporting an electron, it also transports the electron as well as proton. When this molecule passes on its electron to the electron carrier on the inner side of the membrane, electron carrier is to whom? Cytochrome B6 or then after cytochrome F. These are electron carrier. When it transports this electron to electron carrier, it removes this H plus ion into the lumen inside, right? The proton is released into the inner side or the lumen side of the membrane. This is the second step, right? Through electron transport system. Third, C, what is given here? This is completed. You understand, right? The NADP reductase enzyme, because it will reduce into NADPH2, is located on the stroma side because it is also present toward the outer membrane. What happens of the membrane? Along with the electrons that come from the acceptor of electrons of PS first, right? What happens here? Protons are necessary for the reduction of NADP to NADPH plus H. Sometimes we said them NADPH2. How many hydrogens are there? 2 hydrogens. So these 2 hydrogens, they also come from stroma, right? These protons are also removed from stroma. So from stroma, what happens? Only protons are removed. No one proton is added to the stroma, right? That what happens? Slowly what happens? The quantity of H plus ion in stroma, it is decreased and decreased. But in lumen what happens? It is increased and increased. That's why after some time what happens? In lumen, concentration of hydrogen atom, it is more, right? And in stroma, it is less. So these 3 step is given. Hence, within the chloroplast, proton in the stroma decrease in number. In the stroma, it is decreased, while in lumen there is accumulation of proton, right? This creates a proton gradient across the thylakoid membrane as well as a measurable decrease in pH in the lumen. Decrease in pH means when pH is high means it becomes more and more acidic, right? Why are we so interested in the proton gradient? Because we know when a proton gradient is formed, then what happens? The proton, it moves from high concentration to low concentration, means move from lumen to stroma. When it moves through a proton pump, then what happens? ATP synthesis. Because when it moves, so it also provides a sufficient amount of energy for the formation of ATP or for the activate of ATPase enzyme, and thus ATP are formed, right? And in this case, what happens when 4 hydrogen atoms are passed from a hydrogen pump, then 1 ATP is formed, right? Why are we so interested in proton gradient? This gradient is important because it is broken down. It is the breakdown of this gradient that leads to synthesis of ATP. Who is responsible? Breakdown of this gradient leads to the synthesis of ATP. How it breaks down? Means proton moves from high concentration to low concentration. That means when proton move from lumen to stroma, what happens? Synthesis of ATP happens. The gradient is broken down due to the movement of proton across the membrane to the stroma from inner side to outside through the transmembrane proteins channel. These are called channel proteins, or it is CF0 and CF1, right? Channels of CF0 of the ATP synthesis. CF0 means what? It is that is intrinsic protein. It is totally embedded inside the membrane, right? And it provides which transport. It provides facilitate transportation. CF0 means facilitate transportation. Means no energy. It consumes no energy, right? And another one is CF1. It is given here. We will read this one. And because it active transport is there, it also consumes energy. What happens here? The ATP synthetase enzyme consists of two parts. One is called CF0, which is embedded in the thylakoid membrane, totally embedded in the thylakoid membrane, and forms a transmembrane channel that carries out facilitated diffusion, which is passive transportation. This is a part of passive transportation of proton across the membrane. The other portion is called CF1. The CF1 and protrudes, that is, it is extrinsic protein on the outer surface of the thylakoid membrane on the side that faces the stroma, right? It is present to the stroma side. The breakdown of the gradient provides enough energy. It provides breakdown of the gradient means when proton transfer from lumen to stroma, right? What happens? It provides enough energy to cause the conformation changes in the CF1 protein of particle or protein of the ATP synthetase enzyme, right? Which makes the enzyme synthesizes several molecules of energy packed of ATP. When 4H plus ion moves from where? From lumen to stroma, then what happens? 1 ATP is formed, right? In old NCERT it is given 2 hydrogen atoms, but it is wrong because this time NCERT correct this one, right? Chemiosmosis requires a membrane. What are the requirements? First, a membrane, plasma membrane, a proton pump, that is our CF0 and CF1, a proton gradient, that means high and low concentration proton, lumen high concentration, outside is low in case of chloroplast, and in mitochondria outside high concentration, inside low concentration, right? And ATP synthetase enzyme. ATP synthase means what? This enzyme is responsible for the formation of ATP. That means 4 things are given. Which one? First, a membrane, second, proton pump, third, a proton gradient, and ATP synthase enzyme, right? Energy is used to pump proton across a membrane. Because energy is used to pump a proton, that it is active transportation, right? And active transportation means it is present where? In CF1. active transport, and CF0, it is passive transportation, right? To create a gradient or a high concentration of protons within the thylakoid lumen, ATP synthase has a channel that allows diffusion of proton back across the membrane. Diffusion, it is a passive transportation. This releases enough energy to activate the ATP synthase enzyme, right? When ATP synthase enzyme activates, it synthesizes many ATP molecules. Along with NADPH produced by the movement of electron, the ATP will be used immediately in the biosynthetic reaction. Biosynthetic reaction means what? Biosynthetic reaction means it is our dark reaction, right? In which glucose is formed, taking place in the stroma, responsible for fixing CO2 and synthesis of sugar, right? This one is called biosynthetic reaction. Which one is used? NADP and NADPH2 and ATP, they are used, because we know our dark reaction is totally dependent on the product of light reactions. And product of light reaction means what? We have three products: ATP, NADP and oxygen. Oxygen is that means oxygen is released into the atmosphere. It is not present, but ATP and NADP, they are used in biosynthetic reactions, that means in dark reactions. So dark reaction continues till the ATP and NADP is available. Okay. Now look here what we read here. A figure is given. This figure. Do you understand this figure? Right? Look what happens here. This one is our thylakoid membrane. Thylakoid membrane, right? In thylakoid membrane, what happens? Look here. This one is outer and this one is inner. O-U-T-E-R, outer, and this one is inner, right? Lamina. What happens here? Look here. This is the lumen. This one is lumen, right? Lumen means inside. And this side it is stroma. What happens here? Look, first this is our PS2. It is PS2, P680. What happens? PS2, we know PS2 it involves in the splitting of water. Now water molecules are split, H2O. It forms half O2 which comes out of the lumen, and this H plus ion, it is accumulated inside the lumen. Slowly what happens? 1, 2, 3, 4, more and more H plus ion accumulate here. Now here also we know here 2 electrons are released, and these 2 electrons, they are captured by electron transport system, right? Through what is here? Here it is pheophytin. This molecule is called, this one is pheophytin, right? Now pheophytin, it transport this electron to first plastoquinone. Look here, this is plastoquinone. This one is plastoquinone. Plastoquinone, it is a hydrogen donor. It is not an electron acceptor. It is a hydrogen acceptor. But here also, it also accepts the electron, right? Now this plastoquinone, 1 H plus, it also release 1 H plus, right? Receive 1 H plus from stroma. It is from stroma and come and forms a PQH2, plastoquinone plus hydrogen, right? This is given here, plastoquinone. Now plastoquinone, when it is transported to another electron acceptor, that is electron acceptor, it is called cytochrome B6F. It is a complex, right? Complex means what? It is made up of cytochrome B6 and cytochrome F, right? Now this cytochrome B6 and cytochrome F, what happens when it transfer this one? So it also release the H plus concentration, that means H plus ion, into the stroma. So 1 H plus come from this side, second H plus come from this side, 2 H plus come. These 2 H plus accumulate inside the lumen, right? Okay. Now this electron, it is transported to plastocyanin. Plastocyanin, it is toward the inner. side, right? Plastocyanin. And this plastocyanin, it transports this electron to PS first, right? And this PS first, because PS first, it is toward the outer membrane. It is located toward the outer membrane. What happens? By using this electron, it either forms NADPH2, NADPH, look here, NADPH in Z cycle, and it is formed with Z cyclic photophosphorylation. It is not 2, it is Z, right? NADP, and this H plus H2, this is H plus and H plus, this come from stroma. So that means what? It also remove one stroma from one hydrogen atom from stroma. This PS700 for the reduction of, it is called reduction state. NADPH reduced. How? By using of stroma, by using of 2 hydrogen from stroma, right? So 2 hydrogen means here 2 hydrogen removed, and here 1 hydrogen is also removed. 3 hydrogens are removed from stroma. But what happens here? Here 2 hydrogen are accumulating inside the lumen. What happens? Slowly what happens? Here the number of hydrogen atom in the stroma is decreased, but number of hydrogen atom inside the lumen, it is increased, right? Okay, when increased means what happened? Here more H plus ion are there, more protons are there. Now here concentration is more and here concentration is less. So a gradient proton, it is because it is proton, it is called proton gradient, right? Now this proton, they moves outside because it is a common sense diffusion a process in which substances move from high concentration to low concentration. So it moves from, here it is a protein, ATP synthase. Now this ATP synthase, it has 2 molecules. One is one protein body, it forms, it is CF0, and this one is CF1. It is given, right? So CF0, it provide facilitate diffusion, and this diffusion, it is a passive process, means no ATP is used. But this ATP synthase, and when it moves from ATP synthase, that is extrinsic protein, this one is intrinsic protein, what happens? It provide more energy, right? And this energy is used, means this energy activate this ATP synthase enzyme, and when ATP synthase is activate, it forms ATP. When it moves, this hydrogen moves from inside to outside, ATP is formed, right? So here how ATP is formed, here how NADPH is formed, it is given here. It is only, why? It is only due to the hydrogen concentration. Different hydrogen concentration means it is due to the proton gradient, right? Now come to the, where are the ATP and NADPH used? These both are used in our dark reaction. We learned that the products of light reaction are ATP, NADP and O2. These are the products, right? But in these products, O2, this O2, it is removed, means it is released into the air. So now only ATP and NADP is left, right? And this ATP and NADP, they are used in biosynthetic. Biosynthetic means in the process of or formation of food, that is in reduction of CO2 into glucose, or we can say sugar. These O2 diffuses out of the chloroplast, while ATP and NADPH are used to derive the process leading to the synthesis of food, right? More accurately, we said them sugar. Which sugar is formed? First sugar is glucose, then this sugar is converted into starch when they are stored. When they are moved from leaves to other part, they are in the form of sucrose, right? And when they reached to the storage region, they are converted into starch. And this is called biosynthetic phase. This one is called what is biosynthetic phase? Means formation of glucose, right? Of photosynthesis. This process does not directly depend on presence of sunlight, but is depend on the product of the light reaction. What product? ADP and ATP and NADPH. That's why it is called light dependent process. Beside CO2 and H2O, this CO2 and H2O, it comes from environment, right? H2O, it is already present in the plant, and CO2 is come from stomata. You may wonder how this could be verified. It is simple. Immediately after light becomes unavailable, right? The biosynthetic process continues for some time and then stop. For some time means till up to the presence of NADPH2 and ATP, right? And when they finish this reaction, biosynthetic reaction is stopped. If then light is made available, the synthesis starts again. When light comes, now once more ATP and NADP has formed. Then this reaction, means dark reaction, black mass reaction, now we said biosynthetic process, it is continuous, right? So what is, how much, that is how much time this it continues? It continues till the availability of product of light reactions, that is NADPH2 and ATP. Because CO2 is present in every time, but NADP, it is till up to the presence of ATP or availability of ATP and NADPH2, right? Can we say the calling of biosynthetic phase as the dark reaction is a misnomer? Yes, it is a misnomer, because it is not dependent on dark reaction, or it is not present in the absence of the light. It is occurs in absence of light. It also occurs in the presence of sunlight, but it is dependent on the product, right? Also occurs sunlight means in the daytime it is also present. Why it is present? Because at that time we have these products, NADPH2, ATP. At that time it is also available. Light reaction is going on, dark reaction is also continuous going on. But in the night, what happens? After some time, when these products are finished, this reaction is also stopped. That's why it is called misnomer, right? Okay, let us now see how ATP and NADP are used in the biosynthetic phase, right? Okay, now we are going to read the dark. We saw earlier that CO2 is combined with H2O to produce CH2ON, that is for carbohydrate or sugar, because we know carbohydrate means what? Carbon plus water, hydra, carbo plus hydra, this is called carbohydrate. One molecule of carbon and one molecule of H2O, it forms carbohydrate. That's why it is the formula CH2ON, right? Or sugar. It was of interest to scientists to find out how this reaction proceeded, or rather what was the first product formed when CO2 is taken into a reaction, or we can say CO2 is fixed, right? Just after World War II, 1942, among the several efforts to put radioisotopes C14, which radioisotope? C14, that is given, to beneficial use, the work of Melvin Kelvin is exemplary. So Melvin Kelvin used radioactive C14. What he used? Radioactive C14 by algal. Which algal? He used which algal? Chlorella. R-E-L-L-A. He used chlorella, right? Algae name is not given here, but you know, since studies led to the discovery that the first CO2 fixation product was not anyone, but it is C3 carbon molecule, right? It is C3 carbon molecule, that is C3 organic acid, and that is our PG, phosphoglyceric acid, 3-PGA, right? It is a 3 carbon molecule. He also contributed to working out of the complete biosynthetic pathway, right? So complete biosynthetic pathway means it is Calvin cycle plus reverse glycolysis. Hence it is called as Calvin cycle. Why it is called Calvin cycle? After his name, means after the scientist Melvin Calvin. The first product identified was 3-phosphoglyceric acid, or in short it is a PGA, as I already tell you, right? Hence this is also called as C3 cycle, right? How many carbon atoms does it have? Because it is 3, how many carbon atoms does it have? It has 3 carbons. It has 3 carbons. That why it is called C3 cycle. And this C3 cycle, it is universal in all the plant kingdom. Scientists also try to know whether all plants have PGA as the first product of CO2 or whether any other product was formed in other plants. Then what happened? Experiments conducted over a wide range of plants led to the discovery of another group of plants where the first stable product is not a 3-carbon molecule but it is 4-carbon molecule, and that's why it is called as C4 cycle, right? Like amaranthus, sorghum, sugarcane, C4 cycle, because this first 4-carbon molecule is OAA, that is oxaloacetic acid. Oxaloacetic acid. It is a 4-carbon molecule and it is called C4 cycle, right? Here also one thing is there, because they have a specific that is anatomy, that is called Crange anatomy is called. CRANZE, Crange anatomy is present. Since then, CO2 assimilation during photosynthesis was said to be of 2 main types. These 2 main types are there, right? C4 cycle and C3 cycle. So those plants in which first product of CO2 fixation is C3 acid, that is PGA, they are called as C3 pathway, and those in which first product was C4 acid, that is oxaloacetic acid, it is called as C4 pathway. These 2 groups of plants shows other associated characters that we will discuss later. Means after this one we are going to discuss. Look here, C3 cycle, it is universal, means it is also present in C4 cycle, right? So here 2 pathways are given, C3 pathway and C4 pathway. One more pathway is also there, that is called CAM pathway, crassulacean acid metabolism. Crassulacean, A for acid, M for metabolism. But it is present in succulent foods, succulent plants, right? Crassulacean, mostly crassulacean. Succulent from plants means they store water, right? Okay, I will discuss you the different cycles, and then after we can get an idea about the difference, right? Of what is the difference between CAM. CAM is also, it is also C4 cycle. It also C4 cycle. C4 and C3 is present. Here in C4 cycle, C3 is also present, and C3 itself is present, right? Okay, now first another point is here, the primary acceptor of CO2. Who is the primary acceptor of CO2? In C3 cycle, it is RUBP, ribulose bisphosphate, right? So it is also called as rubisco, ribulose bisphosphate, carboxygenase and oxygenase, because it can bind with both oxygen as well as carbon dioxide. Affinity, carbon dioxide affinity is more, right? And they are totally competitive for their active sites, right? That is totally depend upon the concentration. Okay. Now, primary acceptor of CO2 who is? Let us now ask ourselves a question that was asked by the scientists who were struggling to understand the dark reaction. How many carbon atoms would a molecule have which after accepting fixing CO2 would have 3 carbons of PGA? Because we know when carbon dioxide fixation, first fixation, after the fixing of 1 carbon dioxide, now we have the result is 3 carbon molecule. That means what? What is the that is number of carbon which binds with. So at least look here. Now here what happens? Here the first, in case of C3, it is 5 carbon molecule, and this is called ribulose bisphosphate. It is 5 carbon molecule. But when we consider the C4 pathway, right? So there is not rubisco. In mesophil cells, there is which one? PEPcase, PEP, and phosphoenol pyruvate is 3 carbon molecule. It is PEPCASE, phosphoenol pyruvate, right? Carboxylase, right? This one, carboxylase. So 3 carbon molecule. In C3 cycle, 5 carbon molecule, and in the C4 cycle, it is 3 carbon molecule, right? So do you understand what is here? How many carbon atoms would a molecule have which after accepting fixing, it is 5 carbon molecule, which after accepting fixing CO2 would have 3 carbon of PGA, right? Because it has only 5 carbons. When it binds with one CO2, now it becomes 6 carbon molecule. Intermediate step is there, in which it becomes a 6 carbon molecule, and this 6 carbon molecule is also splitting, and then 2 molecules of 3 carbon molecules are formed. 2 molecules or 2 moles of 3 carbon molecules are formed, right? That means we can say 3 PGA is formed. The studies were unexpectedly shows that the acceptor molecule was 5 carbon ketose sugar, that is called ribulose bisphosphate. That is called ribulose bisphosphate. 5 carbon ketose sugar, ribulose bisphosphate, RUBP. Did any of you think of this possibility? Today we know, but at that time it is very hard to think possibility. If 5, that is our 5 carbon ketose sugar, if it binds with carbon dioxide, what happens? It becomes 6. Then how it is 3 carbon? Today we know it is split and it forms 2 molecules of or 2 moles of 3 carbon molecule, that is PG. Do not worry, the scientists also took a long time to conduct many experiments to reach this conclusion. They also believe that since the first product was C3 acid, the primary acceptor would be a 2 carbon compound. They think like so, or they spent many years to trying to identify the 2 carbon compound because they did not understand. So they only think it is that most possibility it is 2 carbon molecules, and when it binds with carbon dioxide, it becomes 3 carbon molecules. Before they discovered the 5 carbon RUBP, right? Okay, it is not very important here. One thing is important. What is this 5 carbon molecule? Now, first cycle is our Calvin cycle. Calvin and his co-workers then worked out the whole pathway and shows that the pathway operated in a cyclic manner. The RUBP was regenerated in a cyclic method, and the RUBP is regenerated, right? And here, what happens? Let us now see how the Calvin pathway operates and where the sugar is synthesized. Let us also at the outset understand very clearly that the Calvin pathway occurs in all photosynthetic plants. It occurs in all photosynthetic plants. That's why it is called C3 cycle. It is called universal cycle, right? Calvin pathway or C3 pathway. It does not matter whether they have a C3 or a C4 or any other pathway means CAM pathway, right? Either they are C3 or C4, but Calvin is present. Calvin cycle is present in all the plant. That's why this Calvin cycle is called as universal cycle. Now, this Calvin cycle, for ease of understanding, the Calvin cycle can be described under 3 stages. First one is carboxylase, second one is reductase, third one is our reduction, that is called, and third one is regeneration. Carboxylation, reduction, and regeneration. What is carboxylation? Carboxylation means one carbon dioxide is bind. So here what happens? One CO2 and one H2O comes and it binds with RUBP, right? And now they form an intermediate element, that is 6 carbon molecule, which after splitting forms 2 mole of 3 PGA, right? Carboxylation is the fixation of CO2 into a stable organic intermediate, right? Carboxylation is the most crucial step of the Calvin cycle, where CO2 is utilized for the carboxylation of RUBP, right? This reaction is catalyzed by the enzyme RUBP CO carboxylase, right? Because it picks the carbon dioxide. So here we write them ribulose bisphosphate carboxylase, and when it binds with oxygen, it is called RUBP oxygenase, right? Combined it is called as rubisco, ribulose bisphosphate carboxylase oxygenase, which results in the formation of 2 molecules of 3 PGA. Since the enzyme also has an oxygenation activity, right, double nature, it would be more correct to call RUBP carboxylase oxygenase, or in short it is called rubisco. It is the most evident protein, or we can say enzyme, in the world. Most. Carboxylation means carbon dioxide fixation. Look here in this figure, very clearly given. Ribulose 1,5 bis phosphate, right? What happens? Start from 1,5 bis phosphate, right? It is called RUBP, right? 1,5 RUBP, right? 1,5 means phosphate, that is phosphorus is attached with first and fifth carbon because it is a 5 carbon molecule. Now what happens here? CO2 and H2O come from atmosphere, and when they bind with this one, this process called carboxylation, right? And they forms two molecule of 3-phosphoglyceric acid, or we can say 2 into 3-PGA is formed. This is the first step, first stable product, right? Now after then reduction start. This one is reduction, right? Here what happens? ATP, how many ATP? 2 ATP and 2 NADPH is used. Now ATP plus NADP is released, right? This one is called reduction. What happens here? After reduction, it forms triose phosphate. Which triose phosphate? It forms PGAL, phosphoglyceraldehyde, right? 3-PGAL, two molecules of 3-PGAL is formed, right? And these two molecules of 3-PGAL through regeneration by using ATP, right? So look here, one ATP, one phosphorus is attached with this one, and they forms ribulose 1,5 bis phosphate. So for understanding here, because we know here 6 carbon are there, but here it becomes 5. How could it understand? For the formation of one glucose means C6H12O6, it needs 6 cycles. Continue 6 cycle. In 6 cycle, what happens? Look here, because it is 5 carbon molecule. Now when we consider this one, it means 5 into 6 means it is 30 carbon. It has 30 carbon. Do you understand? Now 6 carbon dioxide, because in 6 cycle 6 carbon dioxide attached with this one. So this 30 plus 6, it becomes, in carboxylation is 30 and plus 6 carbon come from this one carbon dioxide, 30 from RUBP. Now it becomes how many? 12 molecules. It is become 36 because it is a 3 carbon molecule. How many present? It becomes 12. It becomes 12 phosphoglyceric acid is formed. 12 moles of, in 6 cycle. In 1 cycle 2 phosphoglycerate is formed. In 6 cycle 12 phosphoglycerate is formed. You understand? Okay. After reduction, what happens? Here also it is because 3, because no ATP is used in this. It is used, but phosphoric acid is not attached with this phosphoglyceric acid, glyceraldehyde, right? So what happens here? Here also 12 molecules of 3-PGAL is formed, right? In 12 PGAL, 2P, that means 2 mole of, we write them 2 mole of 3-PGA. It is used in the formation of sugar, right? Means 3-PGA means what happens? It is 3 carbon molecule. 2 mole. If 2 mole is used, means it becomes 6C6, right? 6 carbon. And remaining 10 are used. 10 into 3-PGAL, right? They are used in the formation of how many? 6 ribulose bis phosphate. So when they form 6 ribulose phosphate, they form 30. This 10 into 3, it is also 30 carbon. Here it becomes 36 carbon. Here also it is 36 carbon, right? In 36 carbon, what happens? 30 come to this side and 2 come to in reverse glycolysis, in which sugar is formed, and 30 come into the regeneration, and they forms 6 molecules of ribulose bis phosphate for 6 cycle. So do you understand this one? I think understand, right? Okay. So here in a single cycle, how many ATP is used? In a single cycle, we know here 2 ATP here and 1 ATP here. It was in a single cycle 3 ATP used. That means in 6 cycle, how many ATPs are used? 18 ATPs are used, right? And how many NADPs are used? Because 2 NADP is used in a single cycle. That means in 6 cycle, how many NADP is used? 12 NADP is used. Okay. Reduction, already I tell you here, these are the series of reactions that lead to the formation of glucose. The steps involve utilization of 2 molecules of ATP for phosphorylation. 2 molecules of ATP is used here, and 2 of NADPH for reduction of per CO2 molecule fixed, right? The fixation of 6 molecules of CO2, what is given here? This step involves utilization of 2 molecules of ATP. Already I tell you, in reduction 2 ATP used, and in regeneration 1 ATP used, right? And also, and 2 of NADPH. How many? 2. 2 NADP and 2 mole of ATP are used for reduction of per CO2 molecules, right? The fixation of 6 molecule of CO2, it needs 12 molecule of ATP and 12 molecule of NADP in only reduction, and 6 turn of cell cycles are required for the formation of 1 molecule of glucose from per pathway. In 6 cycle, how much? 2 ATP means it becomes 12 ATP, and 2 NADP means it also becomes 12 NADP, right? Regeneration, in reduction we need 12 ATP and 12 NADP. In carboxylation we need no ATP, no NADP, but we need CO2 and H2O, raw materials. In regeneration, what happens? In regeneration means of CO2 acceptor molecule RUBP is crucial if the cycle is to continued uninterrupted. Crucial means continuously RUBP is reformed, means regeneration of RUBP is continuously we need when we want to continue process, right? Then what happens? The regeneration step require 1 ATP for phosphorylation to form RUBP. How much? 1 ATP. We need 1 ATP, right? So 1 ATP in regeneration, 2 ATP in reduction. That means in one cycle we need 3 ATP. That means in 6 cycle we need 18 ATP, right? In one cycle we need 2 NADP in which step? In reduction, right? Carboxylation no NADP, regeneration no NADP. That means what? In one cycle we need 2 NADP. That means in 6 cycle we need 16 NADP. Correct? Hence, for every CO2 molecule entering Calvin cycle, 3 molecules of ATP, 3 molecules of ATP and 2 NADP are required. It is probably to meet this difference in the number of ATP and NADP used in the direct reaction that the cyclic phosphorylation take place. Now you understand where ATP and NADP are used. NADP and ADP, they are used in reduction, right? They are used in reduction. Second phase. To make 1 molecule of glucose, 6 turn of cycles are required. Because 6 turn of cycle requires. Work out how many ATP. Because in one cycle, how many ATP? We need 3 ATP. In 6 cycle, how many ATP? We need 18 ATP, right? And NADP is a molecule. NADP means we need 2 NADP. In 6 cycle, we need 12 NADP. It is given here, right? To make a 1 molecule of glucose through the Calvin pathway, this is correct. It might help you understand all of this if we look at what goes in and what comes out. The input and output. Input means 6 CO2 comes in and 1 glucose molecule is formed. 18 ATP come in and 80 ADP are formed. 12 NADPH come and 12 NADP goes out, right?